So our first speaker is Kelvin Wong. Kelvin is a PhD student at UCL and is working with Keith Butler on using machine learning for the analysis of small angle X-ray scattering data. As you can see, his talk is on this very subject. So Kelvin, if you could share your slides.
Thank you. Thanks, John, for the introduction. And good afternoon to everyone. So today I'll be presenting on the work that we have done on analyzing small angle X-ray scattering data with neural network enhanced Monte Carlo sampling. So just a brief introduction to the SAXS technique. Small angle X-ray scattering is a technique which gives information on structures at the micro and nano scale. And we typically obtain these kinds of data by irradiating a sample with X-rays and then collecting a 2D scattering pattern on a detector, which we then radially average to get a 1D scattering profile, which can be used for further analysis. So these kinds of analysis are typically carried out using existing software packages such as SAXSView and SAXSFit, and these packages perform nonlinear least squares fitting to fit experimentally obtained data with parameters in scattering models. And these parameters are related to either the form factor or the structure factor of a SAXS profile. So form factor parameters are parameters which are related to the size and the shape of individual scattering entities. So for example, spheres of different sizes will give different SAXS profiles, and basically the spheres and ellipsoids will result in different kinds of SAXS profiles. And of course, if one day synthetically we're able to make nanohards, I'm sure that will give different kinds of SAXS profiles as well. So structure factor parameters are related to interparticle interactions, such as charge and Debye length. So basically aggregated colloid samples will give different kinds of SAXS profiles compared to dispersed colloid samples. So in recent years, several groups have basically explored the use of machine learning for the analysis of SAXS data to perform form factor fitting. So the first example I have is basically a multi-layer perceptron was used to predict the molecular weights and maximum intraparticular distances of nucleic acids and proteins. A software package known as CREASE, which uses genetic algorithms with a neural network surrogate, was also used to basically fit form factor parameters of experimental SAXS profiles. And lastly, and very recently, a multitask model which performs both classification and parameter regression was trained on a dataset containing 1.1 million SAXS profiles spanning across 55 scattering model classes and 219 structural parameters. So what we tried to do was to basically use machine learning to obtain structure factor parameters of a SAXS profile. So this is different from the work that was discussed in the previous slide because we're looking at structure factor parameters rather than form factor parameters now. And we sought to demonstrate a proof of concept for this work using DLVO interactions, which basically describes colloidal interactions as the sum of van der Waals attraction and electrostatic repulsion. So these interactions are mathematically summarized by the three equations on the slide, where basically there are two unknowns in this set of equations, which are basically the charge and the Debye length. So on a short but relevant sidetrack, the DLVO theory does not consider other types of interactions, which are also important at the nanoscale. So some examples of these include non-DLVO interactions, such as repulsive steric interactions and hydrophobic interactions, which play a key role in biomolecular self-assembly processes, such as protein folding. So we performed Monte Carlo simulations to generate data to train basically a model. And we generated 250,000 parameter combinations of charge and Debye length and used them in Monte Carlo simulations to obtain corresponding SAXS profiles. So these simulated SAXS profiles were then used to train a neural network, which takes a SAXS profile as an input and predicts colloidal interaction parameters. So we found that the neural network that we trained was able to predict values for both the parameters with less than 20% error on a test set containing 25,000 SAXS profiles. And at this point, we were happy with the performance of this model, which we call the forward model, because it goes from curves to parameters, and this model basically enables the direct analysis of experimental data. However, we realized that basically SAXS profiles are not unique and that different combinations of parameters can result in near identical SAXS profiles. And in this case, we need to consider a set of solutions rather than a single best fit solution. And therefore, we turn to a sampling technique called Markov Chain Monte Carlo sampling, which allows for the estimation of the parameters as well as the associated uncertainties of the estimates. So basically how this sampling technique sort of works is that we first start by making an initial guess of the parameters, and then we evaluate the likelihood of this pair of parameters being the desired solution by putting these parameters into the Monte Carlo simulations and then obtaining the corresponding SAXS profile, and then we evaluate the likelihood basically using the chi-square function. So basically then we take a random walk and basically we evaluate the likelihood of the new pair of parameters, and we accept this move if the step results in an increase in the likelihood. Otherwise, we reject the move. And basically, if you do enough of these random walks, you can eventually get the posterior distribution of the parameters. So while this is actually good for basically trying to get the distribution of the parameters, it is not directly implementable because it takes approximately 12 hours to generate a single SAXS profile from a Monte Carlo simulation. So basically, if we try to implement this, the entire workflow would take days or months or years to basically converge. So instead, what we did was we used the same data that we had, but this time we trained an inverse model, which goes from parameters to curves. And we use this model to act as a surrogate for the Monte Carlo simulations within the Markov Chain Monte Carlo sampling framework. And the inclusion of this neural network surrogate reduces the amount of time required to carry out the sampling from basically from days or years to minutes. So we use this sampling technique with the neural network surrogate to analyze experimentally obtained SAXS data of gold nanoparticles. And the corner plot on the left of the slide shows the results of the MCMC fitting, which basically shows you the pair of parameters with the maximum likelihood, also known as the maximum a posteriori estimate, as well as the prediction confidence intervals. And we also validated the performance of the Markov Chain Monte Carlo sampling by comparing the fits we obtained from this sampling, as well as the forward neural network and the experimental data. So in conclusion, we demonstrated a proof of concept for using neural networks to predict a SAXS profile given the colloidal interaction parameters and vice versa. We also developed a Markov Chain Monte Carlo sampling algorithm with a neural network surrogate, which is capable of analyzing experimentally obtained SAXS data. So future work includes the incorporation of this MCMC workflow within a autonomous lab to guide the exploration of synthesis parameter spaces. And of course, there is also the potential of using this MCMC workflow as part of a data-driven approach to explore the fitting of other non-DLVO interactions at the nanoscale. So with that, thank you for your attention. I'll be happy to take any questions. And if you're interested in anything that I've talked about today, if you could scan this QR code, which will bring you to basically the paper that we published on what I've just described.